What's up, botanists? We are here to talk about one of my personal favorite subjects, endosymbiosis. And we're going to talk about it, namely, in the light of the origin of chloroplasts today, although endosymbiosis is something that has happened multiple times in the eukaryotic lineage, and we're going to talk about a couple of those times. Here's a list of vocab. As always, pause if you'd like. Whoops. All right. So this is a cladogram that shows a lot of different groups, really diverse. Wow, restart. Here is a giant cladogram showing all of life on Earth as we know it. And we basically split it into three domains. We've sort of talked about this a little bit. Bacteria and archaea are both in the prokaryotic lineage, and then eukaryotes are all the other things, including us and fungi, plants, algae. Well, the photosynthetic pigments that we were talking about last week were in light of the Cyanobacteria, who first evolved photosynthesis. They sort of invented photosynthesis, if you will. Yet somehow plants have the ability to photosynthesize as well. Well, there's a reason for that, and that's what we're going to talk about. The primitive eukaryotes were absolutely not photosynthetic. These were heterotrophs, meaning they were sort of ingesting food, if you will, more or less ingesting it. And they were, in other words, they were not autotrophic. They were not producing their own food. They had to get it from the outside. That's what heterotrophic means, sort of other feeder. And phototrophs are autotrophic. They are sort of self-feeders. They make their own food. Well, these primitive eukaryotes, they had a non-rigid membrane, unlike their prokaryotic ancestors. These were also unicellular, like their prokaryotic ancestors. And so this right here, this is just sort of a diagram of just showing that the outside membrane of the primitive eukaryotes was sort of flexible, and therefore they could ingest things. And if they were to do that, they would do it via this process called endocytosis. I don't know why I'm using past tense. This still happens in unicellular eukaryotes absolutely every day, all the time. And this is a way for these organisms to get particles inside of their body. They sort of are able to use their own membrane and wrap around some particles that are outside of their own body. They wrap around it, and then they are able to sort of pinch off a circular sort of enclosing. They sort of capture these particles from the outside and bring it into their body, and then they're able to sort of deal with whatever they're going to do with it with their body. Maybe they're going to try to digest it. Well, they can deal with getting proteins in and out of that small vesicle that is now inside of their cellular body. Anyway, this is called endocytosis. This is a way for them to sort of get maybe food or any sort of other particle inside of their body. This is not endosymbiosis. Endosymbiosis is the point of today, and I'm showing you something that's not that. Why am I doing that? Well, this is still very, very important in light of endosymbiosis. The fact that they could do endocytosis was a precursor to an endosymbiotic event. So what am I talking about? Well, here's what I'm talking about, sort of. This big blue thing here. Oops. This big blue thing on the left. This is let's just say some primitive, original heterotrophic eukaryote. Okay. And over here, elsewhere, nearby in the water, this is some original photoautotrophic bacterium. So, in other words, a cyanobacterium. They're minding their own businesses on their own, but they're in close proximity, and the eukaryote happens to be sort of predaceous, and it engulfs it in a bacterium every once in a while. It's great, because this bacterium is full of sugars, right? It does photosynthesis, and so it's got sugars inside of it. And that's what it's interested in. And so it will sort of digest this, if you will. It'll sort of ingest it via endocytosis, and then it'll go through some protein digestive type of processes to really break down that bacterium. Of course, the bacterium dies in the process, and all of its nutrients from its dead body are incorporated into the eukaryote. It's great. It's just like very similar to how you and I eat. Except maybe we don't eat our prey live like that. Well, of course, they just have babies. That's all I'm showing here. They don't really have babies. They go through mitosis, right? These eukaryotes will divide into cells. Fantastic. Well, over millions of generations, this relationship evolved between these two organisms, between that original eukaryote and this photoautotrophic bacterium. What happened was it would ingest them, but it would not digest them. So this eukaryote, now it has these little cyanobacteria inside, and they're living inside of it. And what this means is the cyanobacteria can potentially still photosynthesize, but now inside of the body of the eukaryote. And so the eukaryote is sort of reaping the benefits of photosynthesis by keeping it alive inside of its body. In a way, farming it inside of its own body. It's really quite genius. And they didn't think of this, of course. What happened was a very incredibly slow process of incorporation of each other's genes into the other one's genomes. Like, they really incorporated and became a single unit. And it's a really special thing. This is the origin of chloroplasts. Oh yeah, so over time, now we're talking about a different scenario. We're talking about a scenario where these chloroplasts persist inside of these cells even after they divide. So now the daughter cells can sort of inherit those chloroplasts. And the chloroplasts can themselves divide and survive and reproduce inside of the cell. Fantastic. Well, endosymbiosis is something that's really well known, but it's a shocker. It's an amazing thing that has happened. And it actually happened in it's happened multiple times in the eukaryotes, and it's happened to you. You don't have chloroplasts. That'd be so cool. That'd be so cool if we could have chloroplasts in our skin cells or something like that. Wow. But we do have mitochondria in our skin cells, and in fact, all of our cells, every single one of them, has at least a few mitochondria inside of them, of our cells. Mitochondria are just another example of the result of an endosymbiotic event. So plant cells have both mitochondria and chloroplasts, both which are the product of endosymbiosis. And it sort of makes sense. Look at them. Look at the shape of the mitochondria. Look at the shape of the chloroplast. It's the shape of a cell. It's the shape of a little bacterium. Cool. So this is wild. This is wild stuff. We know evolution is happening in a linear fashion, but what happens here is there's sort of this horizontal jump, right? Some eukaryote engulfs and incorporates a bacterium, and they become one. And this really leads to a pretty incredible radiation event, because now this eukaryote has the benefits of being photosynthetic. So what endosymbiosis really means, like when you break it down, when you literally break the word down, endo means inside, sym means together, like symbiosis, and then bio literally means living. So this is a relationship between two species in which one symbiont lives inside of the other. And it has occurred multiple times over evolutionary history, as we'll see. So the current model of evolution for cyanobacteria and plants, which is of course the point of this class, these are plants are the descendants of primary endosymbiosis. So when we use that word plant, we really specifically mean a descendant of primary endosymbiosis, right? Plants did not invent photosynthesis on their own. No, they just sort of farmed the products of it using a cyanobacteria, and that is super, super successful. And all plants, virtually all plants, still use this process. And many other things too, like algae, as we're going to talk about. So So they think it happened right around here, right in between the common ancestor of some primitive heterotrophic eukaryote and the ancestor of a glaucophyte. So glaucophytes were the very first group of organisms that had a cyanobacterium that was photosynthetic inside of them. And this was the very first primary endosymbiotic event. And so just note this little note over here. And that note. Cool. So how do we know that this even happened? Sorry for the squeaking. My dog in the background. How do we even know that this happened? What is the evidence? Well, there's a few things. Cyanobacteria, which, by the way, a common name for cyanobacteria is blue-green algae. Some of their basic characteristics, because, by the way, they survived. The cyanobacteria didn't just get engulfed and then they're gone. Cyanobacteria are still alive. Cyanobacteria are still alive. And when we look at the cell wall of a cyanobacteria, we see something, and ignore the left side here, that's gram-positive, which is just that there are these two types of bacteria, essentially. There are gram-positive and gram-negative. Really doesn't matter what it means for our purposes, but you've got potential to learn about all of that in future biology classes, if you'd like. All that we need to know is that our cyanobacteria are gram-negative, and what that means for us is we've got this, see this lime green line in the middle here? This is peptidoglycan, and this is a major component of the cell walls of bacteria, all types of bacteria. And it's sort of sandwiched in between these two layers of sort of another type of membrane in gram-negative bacteria, such as cyanobacteria. So peptidoglycan is this major component of the cell wall. It's really just found in cyanobacteria, okay? I mean, in bacteria. But the important thing is that cyanobacteria has these additional two walls. Great. And so this is what is able to give us evidence. So, okay. The Glycophytes, Glycophyta, by the way, it's a phylum. I know I've been saying phylum and genus a lot, and we haven't talked about that. I'm going to bring that up at some point soon. But right now, got to focus on endosymbiosis. So this is a glaucophyte, and for our purposes, that means the same thing as Glycophyta. And like I said, glaucophytes were the very first organisms that were that sort of were the result of endosymbiosis, and so there was a cyanobacteria sort of living inside of them, and well, their chloroplast, if you will, it actually has peptidoglycan in the cell wall. And that's actually why we don't call the chloroplasts of glaucophytes chloroplasts. We call them cyanelles. They're not true chloroplasts, because true chloroplasts, which we find in algae and plants, they have lost that peptidoglycan cell wall. And so it's present in this picture that's labeled glaucophytes. It's not, that white line in the middle here is not present in this picture down here. It does, because they lost that peptidoglycan cell wall. Now that being present in the glaucophytes, it's very convenient because it provides evidence for the fact that this cyanelle truly is a cyanobacterium that is just highly, highly modified into being literally an organelle. Okay, so this is a glaucophyte. Cyanophora, this is a genus, so there's a few genera within the glaucophytes. Sorry for the squeaking. And here's what their cyanelles look like. They generally have two, at least in this genus. And they sort of look like these eye spots. Okay, Cyanophora, they also have these sort of flagella, whip-like structures that help them move around. These are unicellular organisms. Very, very primitive organisms. They literally will just have these two. It's like, that's it. That's their whole life. They've got a nucleus, they've got some other organelles in there. What's really important is that the walls of these green structures have peptidoglycan in them, just like cyanobacteria. Okay. And then, like I said, the algae and plants, they do not have that cell wall. And instead, they have this space. So remember this figure from our photosynthesis lecture? Well, oh, I'm sorry, that arrow doesn't point quite to the right place. Oh, I guess it does. It's pointing to this label, inner membrane of chloroplast versus outer membrane of chloroplast. Well, that's the inner and the outer membrane of that cell wall of a gram-negative bacterium that used to have peptidoglycan in the middle. Well, the peptidoglycan is gone, and now they have this space, and I think that's convenient for them because it might allow them to sort of move particles across these membranes more easily. Cool. So there's some evidence that this happened a really long time ago, as you can imagine. And if you notice on this figure here, we've got billions of years ago, the cyanobacteria sort of evolved and radiated, around 1.9 billion years ago or so. And that is represented by this one right here, this line right here. And this wide space of this line correlates with that because that's approximately when the evolutionary radiation, sort of the diversification and kind of large numbers of these organisms were in existence during this time. So then time went on, and folks believe that the first primary endosymbiosis event occurred maybe approximately 1.75 billion years ago. And that corresponds with the beginning, approximately around the beginning of the evolution of eukaryotes. So here we've got unicellular eukaryotes in this category here. Okay. So here's just sort of a review figure from what we've been talking about. Primary endosymbiosis of a cyanobacterium. That process is labeled right here at stage number 3. E is a cyanobacterium. Up here, this is actually some other bacterium, non-photosynthetic, that resulted in all of our mitochondria after a different primary endosymbiosis event by some primitive heterotrophic eukaryote. And so a chloroplast is the result of a heterotrophic eukaryote engulfing photosynthetic prokaryotes. Cool. Well, you ready to have your mind blown? There's this other thing called secondary endosymbiosis. What? This is when, again, a heterotrophic eukaryote, not an autotroph. So something like this organism that just has a mitochondria, something like D. This heterotrophic eukaryote engulfs not a bacterium this time, but it engulfs a photosynthetic eukaryote. What? So it engulfs something that has already undergone the primary endosymbiosis of a cyanobacterium. So here in stage 4, which is this secondary endosymbiosis, it's engulfing this eukaryote that already has a chloroplast, which was a cyanobacterium. Wow. Sweet. And then now you've got this structure, this organism that it has a nucleus. That's, by the way, that's what this pink is. It's got a nucleus, it's got a mitochondria, and then it's got a chloroplast that has an extra membrane around it because it is the result of a secondary endosymbiotic event. Why? Wild. Wow. Believe it or not, there are actually things that have undergone tertiary endosymbiosis, and yes, that would be when another heterotrophic eukaryote engulfs something like this, something that has already undergone secondary endosymbiosis. Oh my god, that's crazy. We're not going to talk about any examples of that. I just wanted to tell you that to utterly blow your mind. Great. So, a little comparison of primary and secondary endosymbiosis. Primary endosymbiosis, we have some outside organism. This one, it's probably just a bacterium because it doesn't have anything around it, and it becomes incorporated into this organism. Well, secondary endosymbiosis, they take this one. They take the result of the primary endosymbiotic event, and then it's right there. Right? Now it's on the outside, and it engulfs that. Okay. Well, some examples of secondary endosymbiosis, or some examples of organisms that have chloroplasts who are derived from secondary endosymbiosis are brown algae, so the Phaeophyta, and diatoms, which are in a phylum called Bacillariophyta. I know that's crazy. I'll have that written down in a slide or two. Diatoms are super cool, and you're going to learn a little bit more about them in your lab, but they are cells that have glass in their cell walls, which is really cool, and they form all sorts of shapes. Okay, well, what we find in different organisms, different photosynthetic eukaryotes, is that, well, we find different colored organisms. Not only different color, kind of looking from a human subjective perspective, but when we look at the wavelengths that are absorbed and reflected, excuse me, in different photosynthetic organisms, we find variation. And that variation comes from the presence or absence of different pigments. And so we sort of briefly talked about chlorophyll in the photosynthesis lecture last week. Chlorophyll looks green to us because it is reflecting green, which would be sort of in the middle here. And so that's what our eyes are seeing. It's absorbing the reds, sort of in the blues and purples. But look at some of these other wavelengths of different photosynthetic pigments. There are these two that are labeled with a FICO. You don't need to know those specific words. You need to know that they're both in the category of photosynthetic pigments called phycobilins. Phycobilins are actually a pretty important group of photosynthetic pigments, as you'll see in a minute here, because they were present in Cyanobacteria. So the phycobilins, for example, they might appear to us to look either sort of more purple or more red, and a lot of organisms have multiple different pigments, and so they might look various colors to us. Okay. And now let's look at which pigments are present in different major groups of organisms, and then see if we can sort of build a cladogram based on those, and or at least track the traits based on these. So here's a small matrix showing the photosynthetic pigments present in various taxa. You actually know some of these. Heteroconts are our brown algae plus the diatoms. Whoops. And the Viridiplantae, this literally means green plant. These are our green algae and land plants. Okay. So how could these photosynthetic pigments have gotten present in these taxa? Well, we need to look at a cladogram for that, so let's build one. Well, the Cyanobacteria have phycobilins and chlorophyll A. So those must have evolved on the way to them. These are all eukaryotes, while Cyanobacteria was our prokaryote. Well, we think the first primary endosymbiotic event was of a cyanobacteria being engulfed by some ancestor of the Glycophytes, right here. Rhodophyta got some chlorophyll D. And so now Rhodophyta is good to go. Rhodophyta, which had chlorophyll A, phycobilins, and chlorophyll D. Well, they got the phycobilins and A from cyanobacteria, and then they evolved chlorophyll D sort of on their own. Now the heteroconts got their chloroplasts from a secondary endosymbiotic event. This secondary endosymbiotic event was the result of a heterotrophic eukaryote ingesting a primitive member of the Rhodophyta, the red algae. But the heteroconts did not have chlorophyll D like Rhodophyta did, and so it's assumed that chlorophyll D evolved after this secondary endosymbiotic event right there on the way to Rhodophyta. But the heteroconts also have fucoxanthin, and they also have lost their phycobilins. Okay, now for the Viridiplantae, that would just be these last three taxa. The kind of true green plants. And somewhere along their lineage, they too lost phycobilins. And they also got a new photosynthetic pigment, namely chlorophyll B. So now if we look at our matrix, we can sort of follow these back on the cladogram and notice that, say, when we look at the embryophytes, which are part of the Viridiplantae, well, on their way, they lost phycobilins, so they will not have that. They gained chlorophyll B, so they will have that. And then since they were the result of a primary endosymbiotic event, we would expect for them to have chlorophyll A as well, but since they lost the phycobilins, they would not have those phycobilins from the original endosymbiotic event. And so to double check the cladogram, yep, it's true. Those are the two they have present, chlorophyll A and B, but not phycobilins. And they also never evolved chlorophyll D nor fucoxanthins. Those were just on the way to the other lineages. And I'll write the common names on here for you to have this as the beginning of your review cladogram. See you next time.